Hey guys, I wanted to give you this sort of slideshow lecture, if you will, to help you get ready for the data set 2, where we're looking at the karyotypes, the genetic patterns. So I'm going to go ahead and switch into present mode. We're going to talk about a couple of things, give you some background, and talk a little bit about human chromosomes and how we can find patterns. And then you're going to apply that and find some patterns to the set of chromosomes in front of you. All right. So this is Lulu the Lioness supplemental slide. So you're going to use this a couple of times. We are not going to go through all of these slides today. So the big question we're trying to answer right now is, how do we determine parentage? How do we decide who the mom of these cubs happens to be? We already made a hypothesis based on observation data, and now we're going to look at the genetics of that situation. So our background, in lion prides, there are often one or two males who join a pride in an area with several females and their cubs. When new males enter a pride, that could be cubs or different males from the area, they displace the resident males, meaning they kind of take the place of the ones that were there before. And they may kill any existing cubs in order to mate with the resident females and produce offspring of their own. So the male lions are not interested in being the father to cubs that don't belong to them. So if you think about the observations you made earlier, is that something you considered? Did you look at how the males interacted? Researchers have been studying lion prides together to gain a better understanding of how their social hierarchy works. And if alloparenting, which is called the caring of young by family members other than the parents, so like aunts and uncles, siblings taking care of other cubs, is present in this species. It is, of course, difficult to determine true parentage for each cub due to the presence of so many possible males and females in a social group. So today, we're going to start looking at the actual genetic data. So our goals or our statements are that I can make observations of karyotypes, which is what we're going to look at today, and reassess our hypotheses regarding relationships. So based on genetic data, would we change what we said about who the parents are? I can identify patterns among the genotypes of female individuals, compare them to the cubs in order to reassess our hypothesis regarding their relationships. So we're going to be looking at genetic information of potential mothers compared to the cubs. So what you have in front of you, these weird looking shapes, is called a karyotype. Karyotypes are organized pictures of chromosomes. And what you have are two of the lion cubs in our pride. All lions are 2n equals 38 and have the same basic karyotype. So the same basic feature, right? You as a human have the same basic karyotype as any other human, same types of chromosomes, and we're going to look at that next. Each chromosome contains the genes, and some of those genes are actually labeled in these pictures. Some, not all, and not on both. So this is sort of what, over here in A, it actually looks like in the body, right? They're not super organized. B, below, shows how scientists kind of separate that out, and they organize them in a pattern. Obviously, we can see this is like largest to smallest. And you can kind of see it from far away. I had a laser point. But we have different shades, different bandings, light and dark, and things like that. There is a close-up of a chromosome, so you can see the details that are not visible. So we see shades of black and whites and grays making kind of different patterns. So let's talk a little bit about chromosomes in general before we start to look at the ones that we have in front of us and answer those questions. So how do we find patterns in chromosomes? So this is the karyotype, or the pictures of the chromosomes of a female human. So these are what yours might look like. Chromosomes come in pairs, right? We see two of everything, and they are pretty much the same size. So the 2 and 1 are the same size, the 2 and 2, and so on. Each of these looks pretty similar. And they also have the same centromere position. So each of these kind of looks like, let me zoom in, an X of sorts. You're going to see a top half and a bottom half. There's a centromere in the middle. So on some of them, it is distinctively in the middle. But on others, it's really high. So that would be where the centromere is. And then they have different banding patterns, the reds and the greens on this one. So we can see, again, different pairs that exist on humans. All humans have 22. The X denotes if you are male or female. So if you are a male, it would be an XY instead of an XX. And then one thing that we have is 21. So 21 is actually the chromosome responsible for Down syndrome. And so this one is a little bit different. You can see that the second one is a little shorter. And so in people who have Down syndrome, they have that extra chromosome. So let's talk about patterns that we could find. So we can talk about chromosomes in terms of how many. So all humans have this many. We talked about how all lions are 2n38. What size are they, right? So if we looked at, again, these are ordered in size. 1 through 6 maybe are kind of big, and then it gets smaller and smaller. We can talk about them in terms of where the chromosome or the centromere is. So like on 1, 2, and 3, it's in the middle, but on 4 and 5 and 6, it's up towards the top, right? So that might be a pattern. How many do they have in the middle? How many on top? And then we might talk about specific banding patterns. So that they have lots of really thin stripes. Number 1 seems to have a stripe on the very top, whereas 7 has more distinct stripes, and 9 only has red in the middle. So how might you describe these things in terms of what you see on the cubs? What's the same? What's different? Do they have the same number? Are they all the same sizes? Where is that centromere on one cub versus the other? Do they have the same locations? When it talks about lengths of the arms, on 1, top and bottom are about the same, but on 4, we have short arms on top, that's the P end, long arms on the bottom, which is the Q end. So you could look at patterns there as well. So those are just some of the options you have. And here are the cubs that we're looking at. So we have cub number 1 and cub number 2. And obviously these images are not great, but we can look at some things. So what patterns do you notice in terms of how many? How many does cub 1 have? How many does cub 2 have? Do you notice if they are male or female, right? Looking at the lettered chromosomes. We can look at size. Do they go big to small, small to large? Do they have, again, that middle centromere, or is it at the top? So for example, if we look at all of the B row, B1, 2, 3, and 4, they all appear to have the centromere at the very top of the chromosome, whereas C1 and 2 are in the middle. And so while the cub number 2 is not labeled the same way, we can look for maybe how many are middle, how many are top. So things like that. These are just small patterns that we're noticing. Right now, you're just looking at a picture of the chromosomes. You don't have the ability to analyze the specific genetic data, which is what we're going to look at on Wednesday in class with data set 3. So again, looking for patterns. Does what you see change what you already believe? So does it change what you think about who the parents are? What new information have we added? So I'm going to go ahead and click into today's worksheet. Oops. There we go. Got to switch over. Let me get to our notebook. So this is the paper that I gave you yesterday. So let's just look and see, again, what those questions are. Since we've talked about what a karyotype is and we've talked about what we can look at, now we're just going to look at what questions are we specifically asking you to answer today about that. All right, so I'm in the field notebook, and you guys do not have a digital copy of this yet. Oh, there we go. I'm going to get rid of that camera. So what happens when you make videos at 10:30 at night. You don't have this slide yet, but you will. Right now, you just have the hard copy. So let me go ahead and pull this up. Here we go. All right, so here is the worksheet. You have the chromosomes, you have a close-up, and here are the questions that we're trying to answer. So on the procedure, it does mention discussing with your group. We're going to start on Wednesday or Thursday with that, but we're going to look at these making sense questions. So what observations did you make? What do they have in common? Why are chromosomes paired? What does that tell you? From where do lions get their chromosomes? So where do chromosomes come from? And how does this happen to produce the pattern you see? How can you use your answer from question 3 to help prove who the parents are? Does this karyotype help you determine the parents of the cubs? And that could be as simple as they have the same parent or they have different parents. And number 6 is probably the most important question, but what other information would be helpful to answer your questions? So again, we're looking at this data here, making observations. We talked a little bit about human chromosomes. It's your genetic data. Think about who you get your genetic data from, and so who the lion cubs get their genetic data from. And again, this is our first introduction into more of the genetics. So a lot of what we're doing in making sense is making some inferences. We're making an informed discussion that we can use to then look at data set 3, which is where you're going to get the actual genotype data, and we'll start analyzing the genetics of the lions. But otherwise, again, look at the chromosomes, maybe expand or change your hypothesis based on some new information, but mostly come in on our next in-class day ready to discuss what we've learned.